Thank you so much for giving me an opportunity to talk. I will be talking about something which is perhaps not so typical in this type of workshops or conferences, but I'm really excited about this project. So let's see how I go with this. Yes, and it will be about epigenetics and particularly what happens in cancer and aging. In reality, these processes are really related, and you can find many publications which relate those two processes when we start talking about the DNA damage, the genomic instabilities, and epigenetics. So this is the work that is done together with, by the way, you also see it like this. Sorry about that. The graphics on my screen, I actually find. Let me see if the rest is the same. Okay, well, here it's fine. So then it's okay. So yeah, this is the work that we're doing together with Tomás Alarcón. He's also based in Spain. Our colleagues from Oxford, which is Helen Byrne and James Davies, he's the experimentalist, and also Marichelle Brunet, she's based now at the University of Edinburgh. So we will talk about epigenetic landscapes and their erosion, but before talking about erosion, we have to start talking about the formation of those landscapes. And after this, I will move to the particular case of the erosion, actually, that happens. So let me start talking about the formation of the landscapes. So in this project, what we do, we want to put two key ingredients, and one of them is the packaging of genetic information, because epigenetic regulation doesn't happen just in the context of a floating chromatin polymer. It happens in this context where chromatin is tightly packaged in the cell nucleus, and this really influences a lot what is going on there. So the initial level of packaging is that you take the DNA double helix and it's wrapped around the protein complexes called nucleosomes, and their epigenetic modifications can occur just at the level of the DNA. By the way, those graphics, they're not exactly the same on my computer. Apologies for this. So the epigenetic changes can happen at the level of the DNA, for example, the DNA methylation, or the post-translational changes that can occur at the level of the histone tails. So at the initial level, when the DNA is wrapped around nucleosomes, you can see this type of the zigzag structure. You can also see it here. This is very typical. This is type of the more open chromatin. And then there are higher order topologies that chromatin can form, particularly loops that can form with the help of additional proteins. And you can even go to higher levels where you can find something that we call topologically associating domains. Those domains, they're characterized by high contact frequency inside of the domain, but the interaction between the domain is rather limited, so they're not connected. If you go even higher, of course, we can go to the level of the entire chromosome. So this is just how the DNA is packaged. And now let's see where the epigenetic comes into place. And as I told you, it can come to as the covalent modifications of histone tails. So you can add different epigenetic modifications to those terminal amino acids. So this is essentially a sequence of amino acids that you can modify. And we will be talking, well, I will focus specifically on these two marks that you can add. This is the 3-methylation and acetylation of histone tails. So depending on the balance between different marks, so the marks can be activating the rather open chromatin, and then the configuration is going to be something similar like this. So here the genetic information is accessible for reading, and then those proteins or whatever, those genes which are encoded there, they can be accessed. Or well, I'll talk about this in a moment, or if there's more of the repressive marks, then what happens is that the chromatin, it starts coiling around itself and it creates those type of structures, and the genetic information which is there is not accessible. So then this is rather repressive chromatin, and the genes there, they will be silent. So if you want for a definition of the epigenetics, essentially it is any change which is not associated with the change of the DNA sequence itself. So you keep it intact. What you control is how the chromatin is packaged. So you either have the information accessible or you have those genes silent. And one more thing that I forgot to mention here that you have in the drawing, that all of those modifications, they're usually mediated by different enzymes that catalyze the addition of these epigenetic marks and removal of those marks. So these are the two key things that we want to put together: how this chromatin fiber is packaged and how these marks are added to histone tails. And this, we want to take those two pieces together and try to reproduce different epigenetic landscapes. And well, I will talk about the rest of it later. So this is type of the model approach that we propose. We decided not to focus on the level of single nucleosomes, because like this you cannot simulate large domains and you cannot see those higher order structures like topologically associating domains. So what we do, we take this chromatin fiber and we are going to split it into pieces of specific lengths of, let's say, some L kilobases. This depends, this resolution will depend on the final data that we would use to calibrate the model. We are not doing this yet. So this is type of the more mesoscopic approach. And then for these two, well, for those genomic sites that we consider, they can be in more compact configuration or more open. And then the epigenetic marks, they added at the level of single nucleosome. There, we consider the modification of two specific sites, which is the Lysyne residue at the site number 4 and Lysyne residue at position 27. There, for the first site, I know it's a little bit of a zoo with the epigenetic marks. It's a bit difficult, but keep stay with me. So the Lysyne residue number 4 can be 3-methylated, and this is going to be an activating mark. And then for the Lysyne residue 27, it can also be 3-methylated, but in this case, it's a repressive mark. But when you add acetylation there, this becomes an activating mark. So those compact chromatin regions, they would be rather characterized by upregulated levels of repressive marks, no, which is this 3-methylation of the Lysyne 27. And for the open chromatin regions, this is going to be the other way around. So the activating marks, they are presented in a higher amounts, no, than the repressive marks. So I told this is part of the epigenetic regulation. I haven't told you anything about the geometry of chromatin. And yes, I forgot to tell you that the model in itself, the way we use it is rather simple, which simplifies it in the way that we communicate with experimentalists, because it's just 6 enzymatic reactions, each of them, because each of the reactions is catalyzed by a different enzyme, no? To remove and to add the marks. So now for the geometry of the chromatin, how it is folded, this is really incorporated in those feedbacks. So the existing epigenetic modifications, they influence which other modifications are added to those sites. So let's say that you have this chromatin fiber, which is folded like this, and then there is this self-reinforcing feedback for the 3-methylation of the Lysyne 27. So then if you have this folding and there is these marks are present here, then this is going to influence, well, this is going to help the addition of the same marks in the neighboring sites just because of the folding. And the way that it's input in the model is in the same way that people obtain this experimental data actually on the chromatin conformation. So imagine you have this contact frequency map very similar to those that I've shown you in the beginning, the way that they obtain it experimentally. I'm just showing you the upper triangular part because the matrix is symmetric. So when you have a high value, it's a high frequency of interaction. So let's say that, for example, this place in here, well, those places in here, those are loops, right? So you just have the folding thing. So the way that model stands is that you get the chromatin conformation as the input, and you want to see what is the epigenetic profile that you get as the output from your model. And one thing that we are specifically interested, also because that hasn't been done before, is the formation of rugged landscapes. So as the cells differentiate, what happens is that some of the genes, they become silent, and some of the genes, they actually activate, right? Because you have this specific genetic signature of what is expressed and what is not expressed. This is the cellular density. So if we talk about epigenetic marks, then you have this alternating pattern of activating repressive marks, activating repressing, no? Corresponding to what genes are supposed to be expressed and which ones are supposed to be silent. So we wanted to look for those rugged landscapes. And the model essentially it does. So we spontaneously get those rugged profiles. So those are activating marks, and this is the repressive mark. And in reality, if you take a look at this configuration here, you can see that it's a circle. Now, so here it's a symmetric problem. Like this, if you run this model, this type of the profile, the pattern, it can emerge in different places. So if I run my model, which is stochastic, and there is small noise in the way that we construct the contact frequencies, and also there is a noise in the initial condition. So this pattern can occur in a different place. So if we consider something a little bit more exciting than just those like locally coiled chromatin fiber with those connections here, and we add we break the symmetry essentially, and we have those sites where you have some loops or connections, then what happens is that this pattern forms exactly the place where you have those non-local connections. This type of sites, they usually called the hypersensitive sites, and they usually correspond to promoters and enhancers, so where the things connect. And it's very typical to see this actually in experimental data, that you have those the activating marks forming there. If I run those simulations several times, you will see that this bump here is forming at the same place. And so you see that there are some fluctuations because the model is stochastic, but it's forming exactly in the same position. So this type of connections essentially make the model more robust, and the pattern is robust. So we can get those racket patterns that we wanted to actually represent the cell, which is more differentiated, let's say. And you can get you can essentially get any pattern depending on the connection. And I can show you the examples of more complex patterns that we can produce, but that's not the focus of the talk. So you can get two of the bumps, so you can get a bigger one depending on what is the chromatin architecture that you're feeding into the model. So this is the rugged profiles that we can reproduce. And now let me finally move to what the talk is about now, about the erosion of these profiles. And for this, we will talk about the hypothesis. Well, I will present you the hypothesis that was proposed by the group of David Sinclair in particular. He has been working a lot about this. He mostly works on aging, actually, but as I told you, those two processes, what happens in cancer is very related to aging as well, because there are many similarities there. And specifically, they were interested in the erosion of those epigenetic landscapes in the conditions of high genomic instability. So, for example, when there are double-strand breaks present in the system. And how they arrived to the hypothesis that they have is that previously it was believed that increased genomic instability accelerates aging, right? So you get the accumulation of mutation, and then this leads to the shortened lifespan. I actually don't have. Okay. Then there was a counter evidence to this. For example, there were experiments with mice and other cloned animals that did have mutations, but they had normal lifespan. But at the same time, the mice with a reduced DNA repair capacity, they experienced accelerated aging without having any of the mutations. So then what they proposed is that perhaps the aging is not the result of the loss of genetic information because you get mutations, but perhaps it's actually the loss of epigenetic information. I'll connect it all back just in a second. And so, for example, more specifically, there are types of enzymes which participate in the epigenetic regulation. For example, there is this certain family of enzymes, and they participate in the removal of the acetylation marks from for example, from the lysyne residue 1227 that we include in our model. So in the normal case, this lysyne just removes those marks, and it controls how much of the acetylation of this site you can have. And if you remember, I mentioned that this mark is the activating mark. So you get so usually when there is a lot of this acetylation of the site, those genes tend to be open and accessible for reading. Then when you have the double-strand breaks, those certain family, they are relocated, so they sequestrate away from the histones, and they are relocated to the repair sites. And because there they participate in the repair of those double-strand breaks. So if the level of breaks is really high, what happens is that those enzymes, they get relocated to the sites of the DNA damage. And then the hypothesis that they have is that, well, actually they proved it experimentally, is that the chromatin architecture is not restored back once the DNA is repaired. So you take away those enzymes, right? And then this influences the system so much that you don't get the same profile when the DNA is already repaired. So this is the same thing that we did. So we have our model with our epigenetic landscapes. And what we did, we just added one more reaction of the recruitment of these enzymes to the double-strand breaks. there is no special coordinate in here. So we just assume there is some level of the breaks that exist in the system. So you just take them away, you relocate them. And just to finally connect it back to what we were talking about on the first day, essentially, this is just connecting those two hallmarks of cancer that we mentioned, the genomic stability and a phenotypic plasticity. Because when you start eroding the epigenetic landscape, you essentially you get the loss of cell density, which is part of the phenotypic plasticity that you get in here. So those processes, I mean, perhaps through this hypothesis and this work, those processes are kind of related, and you get one emerging after the other.
So where's the genomics here?
The double strand breaks, the breaks,
Just breaks.
well, yes, exactly.
Not mutations.
No,
Which radiation used, or it's randomly.
it can be just run. I mean, you don't. Well, you know that, for example, the DNA repair, it happens a lot in aging and in cancer. It happens all the time. I mean, I don't talk about what causing this, but we know that this is occurring. We know that there is genomic instability in cancer for sure, in aging as well. It is just there. Yeah, but we do not include this part in the model.
I'm sorry, the pink enzymes are which ones?
Say it again, please.
Yeah, the pink enzymes, they're the
This
ones
guy.
that would otherwise do what?
Those are the guys those are the certain families. So those are the ones that remove those activated marks from the chromatin. And they as well, they also participate in the repair
So they
of
have
DNA.
really different functions.
They have different functions apparently. Yes, but and it's not the only family. There are also other types. Well, it's not only sirtuin family. There are other enzymes that also participate in epigenetic regulation, and they also participate in the DNA damage. Everything is a little bit related there, but I can find you the names of the enzymes if you're interested. But anyway, so we added this part into the model, and then we try to see if we can get the erosion of epigenetic landscapes. So here I'm just going to show you the simulations where we upregulate the amount of the DNA damage that exists in the system like tenfold. And you can see that here, this is very similar to what we had before. So we still have this profile. But now if I start repeating those simulations, you see that the profile that was very robust before, because I had this chromatin conformation in here, it moved to another place. So now we don't have the robustness of the system. So indeed, what we get from here is that the topology of the chromatin folding influences what is going to be, what is going to happen. So not all of the chromatin topologies are the same in terms of the robustness. Some genes, they can, for example, lose their structure and they get the erosion of the profile, and some of them perhaps not. I mean, let's see. So if I repeat the same thing for a more complex geometry that we have here, and I repeat this over and over, in reality, you get the same profile. So this type of the chromatin conformation is more robust than compared to this one. But however, if I keep increasing the amount of genomic instability, amount of DNA damage that exists in the system, so essentially all of this goes, so there is no pattern any longer. And what we see present, it's a uniform profile of activating marks. So essentially you get the erosion of this profile. So everything starts being activating. And this is the same thing that usually happens in cancer cells and also during aging, because you start seeing things that, so the genes that are supposed to be expressed, they're not expressed, or they express less, but then you also start seeing genes which are not supposed to be there, right? And they appear because you get the erosion of this landscape, and then the chromatin changes its folding, and then you get access to something that you're not supposed to be seeing. Or for example, well, there are changes in the epigenome that eventually result in what you see in the cellular function. And well, that's it. So, well, I forgot about the disclaimer here. That what we do so far is this type of program where you start with the chromatin conformation and you do a forward problem of running the epigenetic model, and then seeing what is the epigenetic profile that you get. The real problem is more complex because you get this thing in a loop, and then you can have other factors like transcription factors influencing what is going on. And in reality, this is kind of this type of thing going on, but we are not there yet. So far we are doing this forward problem of taking the conformation and seeing the epigenetic profile, and just preliminary conclusions that we have and what we are doing right now. So we indeed get the formation of rugged epigenetic profiles in the context of the chromatin, which is folded, not just in a stretched line of a chromatin, which was something that was new. then indeed we can see that we confirm that using our model we can provide a mechanistic explanation how this relocation of certain enzymes can destroy those rugged epigenetic profiles. And several things that we have is that right now we're working with the data to confirm the formation of these bumps that are formed at the hypersensitive sites, or the sites where you have those non-local connections. So here we have the data. But another thing that we wanted to try, and here we do not have the data, is to try to infer back the connectivity back, so to do the backward problem and to try to infer what is the chromatin conformation for the perturbed epigenetic profile of normal versus cancer cells in different types. So we're trying to find the data for this thing, but not there yet. And then, yes, of course, trying to work on this loop issue that I talked to you about, not just a forward problem, but try to see how the epigenetic landscape influences the chromatin conformation. So the second part of the feedback. And that's it. And those are the people that work on this project with me. Thank you so much.